Hello dear students, today we are going to see part 3 and this is the last part of our chapter chemical reaction how far how fast. This is for IGCSE Cambridge and this is Mayank sir from the new Tulip International School. Now in this part we are going to learn about enzymes. In last session we have seen about catalyst. So catalyst is the chemical term while enzymes are specifically related to biology. So enzymes are protein molecules produced in living cells and they are catalysts which speed up hundreds of different chemical reactions going on inside the living cell. And these biological catalysts are very specific in that each chemical reaction has a different enzyme. So these enzymes are very specific for their chemical reaction. Remember this thing very carefully. And there are literally hundreds of different kind of enzyme in our body also, and enzymes all have an active site. These enzymes are made up of protein and these enzymes have active site. Now what is this active site? Active site is actually the binding site with which the substrate will bind with the enzyme. So that particular site is known as active site. So the active site is a particular shape and locks it into a corresponding shape in a reactant molecule. So when these has happened, the enzyme can work to break up the reactant. So here lock and key model type of reaction will be there with enzyme and substrate. However, you can see that every lock has specific key. Okay, lock cannot be opened by any other key. It is the same thing over here, that every reaction, every substrate has a particular enzyme. So here is the diagram for lock and key model understanding is given over here. The enzyme molecule which are shown in red, pink, green or blue color lock on exactly to a particular reactant molecule. Reactant molecule are yellow in color. So once the enzyme is locked on, the reactant molecule break up into smaller pieces. For example, now we will take one example where enzymes are actually working. So for example, hydrogen peroxide is produced within our body. So however, it is extremely damaging and must be decomposed very rapidly. So catalase is the enzyme which convert hydrogen peroxide into harmless water and oxygen within our livers. So this in our livers catalase enzyme is there that will convert hydrogen peroxide into water. Although many chemical catalysts can work under various conditions of temperature and pressure, as well as alkalinity or acidity, biological catalysts operate only under very particular condition. So for example, they operate over a very narrow temperature range, and if the temperature becomes too high, they become inoperative. So these enzymes are very specific for temperature and pH also. At temperature above about 45 degree, they denature. These means that the specific shape of the active site of enzyme molecule change due to breaking of bond, and the enzyme will get denatured. Now there will be no more functioning. These means that the reactant molecule are no longer able to fit into the active site. A huge multi-million pound industry has grown up around the use of enzyme to produce new material. For example, biological washing powder contain enzymes to break down stains such as sweat, blood, egg, and they do this at a relatively low temperature, that is 40 degree. These reduces energy cost because the washing uh, water does not need to be heated as much. They will work at lower temperature. Other industrial process also make use of enzyme. First, it is in the manufacture of baby food. Enzyme called proteases are used because it requires pre-digest food, and this is because young babies are unable to digest protein molecules. So for pre-digestion, proteases are added to baby food. Now second is the enzyme isomerase is used to convert glucose syrup to fructose syrup, because fructose syrup is much sweeter than glucose syrup and can be used as a sweetener in sliming food as less is needed. And in the production of yogurt, milk is initially heated to 90 degree for 15 to 30 minutes to kill any bacteria in the milk. After cooling to 40 degree, a starter culture of Lactobacillus bacteria is added, and the mixture incubated at 40 degree. Incubated means will kept at 40 degree for 8 hours. So the bacteria, the same process by which we are making the curds. So the bacteria ferment the lactose in the milk to lactic acid, which causes the milk protein to become solid. That's why curd will become semi-solid while milk is liquid. So lactobacillus bacteria is there, the name should be remembered. So here is a picture is inserted in the slide, that cheese is separated into curd and whey by the addition of bacteria, and the liquid whey is separated from the curd, which are then pressed. So this is the cheese making industry. You can see the picture. Now in industry, enzymes are used to bring about reaction at normal temperature and pressure that would otherwise require expensive condition and equipment. So enzymes can make the reaction friendly to us, and successful processes using enzymes need to ensure that the enzyme is able to function for long period of time by optimizing the environment. Second, enzyme is kept in place by trapping it on the surface of the inert solid. This is a very important technique. If you want to use the enzyme again and again, then you can take a plastic sheet or acrylic sheet, and in which you can trap the enzyme on that sheet. So what will happen, the active site should be open in this case. So whenever you want to carry out any reaction, you can just insert this sheet into the substrate, and after the reaction get over, you will take this sheet back and again store it. So what will happen, the enzyme will remain unchanged, enzyme will remain unaltered, and you can use these enzymes again and again. And third is continuous processes occur rather than batch processes. अभी any fermentation process or any other conversion process can be happen in two type, either batch process or continuous process. So continuous process will be more useful because the enzymes can be used again and again in that thing. So if any doubts, you can contact me by WhatsApp, students. Thank you. Bye bye.